Hello everyone, my name is Pierre De Langen. I'm a PhD student, so I'm really happy to be here to present our work. So first of all, I'd like to thank the RegSys committee for letting us presenting our work on intergenic transcription. So short introduction. In transcriptomics, usually most of the work is focused on the genic regions that produce abundant RNAs that are then translated into proteins. But the intergenic regions of the genome also go under transcription by the RNA polymerase 2, which I will be referring to POL2. And it also produces mostly unstable, but also shorter RNAs. But these RNAs were found to also be essential to cellular identities. So, for example, it was found that some long non-coding RNA were essential to preserve the pluripotent stem cell identity. And in genome-wide association studies, the majority of trait-associated variants or disease-associated variants do lie in non-coding region of the genome and not in exons of the genome. And an aberrant expression of these non-coding elements was also found in several diseases, such as cancers. So in this work that I will going to present you, we only focus on these intergenic regions of the genome. And despite a much weaker signal, we will integrate a lot of data to overcome this weak signal. So the three main topics that I will present you today is that how do we created an atlas of binding site of RNA polymerase 2. How did we characterize it by tissue specificity, and also our study of intergenic transcription in cancers. So how do we transcribe this likely transcribed region? Previous studies mostly targeted the transcripts themselves, such as the phantom 5 project that use the cage. But most of these transcripts, as I said, are mostly unstable. So we go one step above and we target the enzyme that is responsible for the transcription, so the RNA polymerase 2. And we use ChIP-seq for that. And as a very short reminder, ChIP-seq aims to find where does the protein binds onto the genome. So what we've done is that we have retrieved every possible ChIP-seq experiment that were available from public databases, such as GEO or ENCODE. So we got 906 of them that were past quality checks. So all of these have been manually and uniformly curated. So this might sound trivial, but this is a lot of work, and this is really required to have high quality data because what's available on GEO, if you take the annotation as is, it's often really bad. And all of these we're gone under uniform reprocessing, so from the raw FASTQ files up to the peak calling stage. So after the peak calling, what we'd like to do is to identify what we refer to as consensus peaks. So for example, here you can see that this is the same binding events. And we get over 190,000 of them in the intergenic region of the genome. And these are robust because we can at least observe one peak in 2 experiments across the 906. So here are 2 other examples. So our consensus peaks are genomic region of the genome. So they are around 200 to 300 base pairs. And as you can see, they often overlap on a regulatory element of the genome, which is what we expect. So what are these regions? And so we compared our POL2-bound regions to reference databases, so originating from ENCODE or Phantom 5 or REMAP, which is a database of transcription factors. And what we found is that we mostly fall into regulatory elements that are open chromatin or enhancer-like. And we have a few promoter-like signatures, but they are mostly located on TSS of long-coding RNAs. And we have around a quarter that overlap bodies of non-coding RNAs. So what we can also do is that we have our POL2-bound regions, which correspond to genomic location. We have our 906 experiment. We've annotated our experiment with an high-level annotation to simplify how to interpret the data because we have over 250 different cell line and tissues, so it's not interpretable as is. So one question that we can ask is that is there some consensus peaks that may be specific, for instance, to the cardiac experiments or to the embryo experiments? And to visualize this, we use UMAP to visualize the POL2 occupancy across experiments. Well, typically in single cell, you will see that the samples or the cells themselves are being visualized with UMAP. Here, one dot corresponds to one POL2 consensus. And here, for example, you can see there is clearly one group that is specific to the embryonic cells, some groups that are specific to blood or immune cell, and one group that is specific to cardiovascular cells. So to retrieve these groups, we use graph clustering, and we identified 51 clusters with very specific identities. So the previous one that we observed was the embryonic one, the cardiovascular one, and the blood or immune one. And some are more ubiquitous, and some are are very specific to other types of tissues. So, for instance, as an example to show that it actually makes sense what we observed, we compared, we used the roadmap chromosome annotation. So, at each of our genomic regions, at our POL2 consensus, we can say in embryonic stem cell epigenomes, what is the epigenetic state? So is it a TSS? Is it a repressed region? And what we've seen is that if we took only the POL2 consensus that belong to the embryonic cluster compared to the other one, we are strongly enriched in active epigenetic states such as enhancer, and we are strongly depleted into a repressed epigenetic state such as repressed by polycomb or quiescent regions. So I talked in the introduction about variants in GWAS, and what we've done is that we used the LD score regression to identify which traits had the strongest heritability enriched in our region. And as we can observe, we have a lot of traits that are related to the cardiovascular system. So, for example, intraocular pressure that is tightly linked to blood pressure or to pulse rate or to heart disease. And similarly, for this cluster, the blood or immune one, we've looked at nearby genes, and we can see that we have a lot of immune-related genes. So all of this seems to make sense, but our approach does not directly identify transcript. So we reanalyzed RNA-seq datasets from 3 consortiums. Sorry, I don't really feel well.